hopefully we are live. We'll see. We'll just check it in a second, seeing if we're connected. So it seems like we are live. So hopefully you can see this. We're about to start our B2 exam revision live stream. Obviously this is a few days before the exam. The exam's not till Monday morning, but obviously because it's Monday, we're doing it on Friday. Hopefully you've still got the picture. Anyway, so we're doing it a bit early, so you've got the weekend. You can watch it several times over, which I'm sure you will, and be able to get all the benefit from it. You can get the slides from our revision website, and also we will put them in the link to the video as well. So this is going to be going through everything in the B2 exam. We're halfway through now. We've done B1, C1 and P1, paper ones. During that time we've covered all the topics for that. Now in those units, technically they can ask you little bits from that because you've already done those and this is sort of following on. So I'm not going to go through the basics in this video, say of cells, prokaryotic, eukaryotic and things like that. I'm going to go into the specifics that are on B2, but they can ask you little bits about that. I would say cells really being the fundamental sort of concepts would be something to have another look at just in case. Things like photosynthesis and respiration, I'm going to talk about those, and they'll be part of it as well. So they might be something that might come up on this. Little bits about them. There won't be massive questions about them like that was for B1. This is B2, but there can be small sections of it. So it's worth just refreshing your memory on those. But this is going to cover the topics that are specific to B2, go through it in a bit of detail, not loads of detail because we don't have time to cover everything, but we'll do what we can. The extra detail you'll need to get yourself. I'm just putting on my timer here so I know how long we've had, and we should be ready to go through the different bits. The email address as always is contactscience@folkestoneacademy.com. Send your questions there. Miss Walker will answer any that she can answer immediately, just over email, straight away. And I've got Mr. Pritchard here with me. If there's anything that's sort of a bit more involved, we'll try to go through it on this board here, and I might go through a few little bits as well to supplement what we've got here. You can access the slides I said on our Academy website, and link in the description hopefully soon. So, going straight into it. The first sort of topic that's covered in B2 is all to do with controlling body systems. There's two sort of aspects to it. One is about nervous response, one is about hormonal response. Nervous response isn't actually too bad, I find. There's just a sort of pathway you've got to remember, and then one sort of specific bit about gaps between neurons, which are the nerve cells called synapses. We'll go into that and we'll look at those next. But overall, you need to know a reflex action. Now, a reflex action is a type of nervous response. There's a normal nervous response and there's a slight difference in it, and I'll highlight what that is when I get to it. But in a reflex action or a reflex arc, it's a response that you sort of do without thinking about it. So you might touch something hot, you might prick your finger or cut your finger on a sharp object, and your body instantly responds to prevent you from further harm. What it is you start with a stimulus. So that stimulus would be me touching a hot object or touching a sharp object. That would be the stimulus. The stimulus is what is causing the response. That then is picked up by receptors that I would have in my body. Receptors, generally you think of your senses, and that leads on to the next bit. But your senses such as apologies for that background noise there. Your skin is a receptor, has receptors in your skin that pick up touch, your nose, your eyes, your ears, your tongue. These are all type of receptors that can lead to a nervous response. That then sends a message down what's called a sensory neuron, or neuron. I remember that because sensory neuron, it comes from your senses, the senses that I went through. So that sends a message down a sensory neuron. Which goes to, now this is where there's a slight difference in a normal nervous response and a reflex action. In a reflex action it goes around a relay neuron. A relay neuron is like a relay race, it just passes the signal on to the next point in the chain. Now the relay neuron happens very quickly, it's a quick response, because if I touch a hot pan I don't want to think about it, go oh that's hot, might be causing me harm, before actually responding. The body responds instantly to protect itself. So the relay neuron passes the signal on to the next bit of the chain and make sure that you respond quickly to the signal. Anyway, the relay neuron passes the signal on to what's called a motor neuron. And a motor neuron, you can remember this, or I remember this, because a motor neuron creates movement normally. It actually is linked to what's called an effector. And an effector, generally in a nervous response, is a muscle. We respond to it in a certain way. So I've touched the hot pan, that's picked up by receptors in my finger, send a message down a sensory neuron to my spine where there's relay neurons which fire the message back to the muscles in my arm and tell me to pull my hand away. So the effector is a muscle which causes a response, i.e. my arm to move, and that happens really quickly. Difference being in a normal nervous response is rather than going through a relay neuron, it actually goes through the central nervous system, the brain and the spine. That's equivalent to you thinking about it. Now, I'm going to pick up this pen over here, you might not be able to see it, because I want to write something on this board. So it sends a message, the stimulus is the pen, sends a message through my eyes, which are the receptors, or receptors in my eyes, sends a message down a sensory neuron to my brain interprets that signal, sends a message back out to the down the motor neurons to the muscles, the effectors, and that causes me to reach for the pen and pick it up. Okay, so just to go over, we have a stimuli, or stimulus, which is picked up by a receptor, which goes down a sensory neuron, and then this is the bit that, as I say, is different depending on what sort of thing you do. It can either go to the CNS, which stands for Central Nervous System, that's the brain and the spinal cord, or if it's a reflex action, it will go round a relay neuron, sends the signal quickly on. Then that goes to a motor neuron, and then that goes to an effector, and the effector can be a muscle or it can be a gland in some instances. Okay, so that's your sort of nervous response, like flow diagram if you like, that's a quite typical one. Now between the neurons there's actually little gaps, and it's important there's little gaps because you don't want signals firing all the time, you want these messages to be sent correctly at the correct place and the correct time and place really. If your nerves were firing all the time, it can lead to poor signals being sent around the body. For example, if you've got damage to your spine, some people feel constant pain where their nerves are constantly firing and it makes their body interpret it as pain. And it's not a good thing to be in because you're constantly feeling a pain, even though it's actually nerve damage rather than anything actually causing you physical pain. So between the neurons there are actually gaps where there's a different signal sent. Now down the neurons, I should have mentioned, is an electrical impulse. That's an electrical signal sent down the neurons at high speed. That happens really quickly. Now these gaps are tiny, so there's only a tiny gap, but when that electrical impulse arrives at the end of one neuron, it actually sends out or causes it to release chemicals. Those chemicals are released and they actually move across that gap where they're picked up by the neuron that's attached to it. When it picks up those chemical signals, it triggers it to send another electrical impulse back down that neuron. So if this was our sensory neuron, it would reach here, the end of it, it would send a chemical signal across to maybe the relay neuron, which would then send an electrical impulse down to the end of that to pass it on to a motor neuron. So in a synapse, the little gap between two neurons, the chemical signal is actually sent, whereas through a neuron, an electrical impulse is sent. That's a slight difference. So that's something they can sort of talk about or focus on, the difference between a synapse and the electrical impulse sent through a neuron. So that's our nervous response. Now, other responses are controlled often by hormones, and the control of like body conditions, all the conditions within the body, is actually called homeostasis. Your body is a finely tuned machine. Okay, it's controlled by lots of different things, and it needs to be kept at a regular sort of pattern of working. Otherwise, there will be problems and can lead to, in some cases, fatal problems to occur, things you really don't want to occur, like temperature, for example. Your body temperature has to be carefully regulated. If it gets too high, you'll be in serious trouble because your enzymes won't work. They'll become denatured. Enzymes we talked about obviously in B1. If it gets too low, enzyme activity will slow down to the point where, again, you'll start to have problems. An important process, as you well know, is respiration. If that can't happen quickly enough, your body can't get energy, all the muscles can't get the energy they need to survive, the heart needs energy constantly to beat, so it will start to cause issues. Homeostasis is the maintaining of those optimal conditions, those conditions for your body to work perfectly, controlled internally all the time. The main ones we talk about, and we need to know a little bit about, are blood glucose concentration, so that's sugar levels within your blood, maintaining them at the correct level. We need sugar in our blood, or glucose in our blood, because we need it for that process of respiration, and we need to keep the levels correct for us to continue to remain active. Body temperature, again, enzymes are very sensitive to changes in temperature, so we need to ensure that the temperature is right for those enzymes to work best. They work best at around 40 degrees, and body temperature around 36.4 to 7, sort of in that range. And we need to make sure it stays in that. That's internal body temperature. External body temperature can vary slightly on that, but the internal body temperature really needs to stay within a small little range, if you like, for it to work correctly. And then water levels. Obviously, if your water levels drop too low, you could dehydrate, which can cause you all sorts of problems, and the body needs to be controlling the levels of water within your system so that your cells remain active and are able to keep functioning. I've talked a lot about us in this example, but just about every living thing has to have some level of homeostasis, keeping internal conditions the same. So there are ways they do this. Often, a lot of these methods in homeostasis are controlled by the endocrine system. The endocrine system is the hormonal control of body conditions. There are glands, you need to know, within the body that secrete, if you see that word in the exam, just means releases or gives out. It's just a word they like to use. So the glands that are important within the endocrine system, you have a gland at the bottom of your brain called the pituitary gland. This is a really important one. It's called the master gland sometimes. It controls a lot of the function of other glands of the body. It's very close to a part of the brain called the hypothalamus, which is responsible for sort of monitoring internal conditions. They can talk about that. So that's responsible for checking everything inside the body is at the right level. And then the pituitary gland is often the gland that will release the first hormones to signal other glands in the body to respond or create other effects in the body. Sometimes they can be nervous response as well, not just hormonal. You've got the thyroid gland, which is particularly of need for you to know if you're doing the higher paper. Thyroid gland, and we'll talk about it briefly within this a bit later. It controls levels of thyroxine, which control your metabolic rate within your body. Metabolic rate is how fast you can get energy into your cells, and that helps with growth. You've got your pancreas, which secretes certain hormones. A particular one with that is insulin, which again we'll talk about. The pancreas controls levels of insulin within the body, and that's important for controlling blood glucose. You've got your adrenal glands, which release adrenaline. Adrenaline is another important one because it leads to the fight or flight mechanism. If your body needs to protect itself from harm, adrenaline will be released into the bloodstream, which will cause a response such as raised heart rate, elevated metabolic rate, things like that, which will cause you to go into a fight or flight mode. With adrenaline, I find it really interesting. They've actually had examples of people who have been in bad car crashes, and they've actually found the person several meters or a good distance away from the actual accident, and the person has no recollection of how they got there. That can be because levels of adrenaline have been released, their body's kicked into what's called fight or flight mode, they've moved away from danger without even being conscious of their body doing it to protect themselves from further harm. So that was the pancreas, the adrenal glands, and then important ones for development of people during puberty are the sex glands, which are the ovaries in a female and the testes in a male. These release two different hormones, obviously depending on the gender. Estrogen released from the ovaries and testosterone released from the testes. These are important for causing the body to develop during puberty, during that time. And you need to be aware of some of the changes that can occur. And then they're also, particularly the ovaries, we will talk about that specifically when talking about the menstrual cycle and about controlling fertility and how that process actually takes place. One of the ones here, I've talked about these specific hormones they can do. Talked about adrenaline, the fight or flight hormone, raised heart rate, metabolic rate, things like that. Insulin, as I said, I'll go into some specifics. ADH is antidiuretic hormone. It actually causes the kidneys to actually absorb or take more water out of the blood or not take as much water out of the blood and controls the water levels within the body. It's antidiuretic hormone, so it allows the water levels to be controlled, and it is one that they can talk about in the GCSE. Now, as we well know, in the GCSE, they do like to bring in other things, and as Mr. Pritchard tells his classes, and a good bit of advice is, don't get hung up on too many specifics. Look at the general sort of patterns that the question's asking about and relate it to something you do know or you do remember. Some of the names they may give you of certain things may be very strange. Do not get hung up on that. Do not get hung up on the examples. Classic from B1 that you all know about is the potato carrot example. Don't get hung up on the specifics. Think about it in general terms. If they're talking about a hormone, think about it, the hormone responses that I've mentioned, and think about how the body responds to it. The important thing about hormones compared to a nervous response is nervous responses are electrical impulses. They happen really quickly because they're sent down your neurons at a fast rate. Hormone responses are slower because they have to travel through your blood, and your blood takes a bit of time to be pumped around the body to where it's needed. So, for example, hormone release from the pituitary gland will take a while to get to, say, the adrenal glands where it may be needed to cause another response, or to the ovaries where it causes another response. So that can take a little bit of time. So it's a bit slower than a nervous response. But the thing with hormones is the effects tend to be longer lasting. A nervous response is instant in some ways. I see the pen, I pick up the pen, that's it, the nervous response is over. Hormonal response takes a bit longer to have an effect, but then generally lasts longer when it is in effect. So I said we needed to know some specifics. Controlling blood sugar is a specific example that you need to know. And I'm talking about things like called negative feedback loops. Now they're often like put on a pedestal and thought that that's actually really complicated to understand. It's actually really, really simple, and I'll come to it in a second. So we need to control blood sugar because glucose, as I said, is needed for respiration, and it's controlled by a hormone insulin, which is released by the pancreas. As levels of insulin go up, it actually causes a response. Glucose is converted to glycogen. You probably can't see it on the board up here because I know it's very bright out today, so it might not be coming up. But if you've gone to our website and got a copy of the slides, then you'll be able to see it there. But anyway, insulin, when it's released by the pancreas, it causes glucose to be converted to a chemical called glycogen, which is here as well. Glycogen is a store of glucose. It can store it in the body as glycogen for when it's needed. It's actually the liver where this transformation takes place. The liver converts glucose to glycogen for storage. So that happens if your blood glucose levels are fixed at a normal sort of level, so it should be like at this point here. If they start to rise, so you've just had a sugary meal, you've just had a chocolate bar, or you've had an energy drink, and those levels start to go up, the body responds to it by actually sensing that in the hypothalamus and then sends a message out for the pancreas to release insulin, which causes the glucose to be converted to glycogen and the levels fall back to where they should be. So that's if the levels get too high, insulin is released or secreted, glucose is converted to glycogen and the levels fall back down. Now, if the levels of blood glucose fall and get too low, the body needs to respond to bring them back up. It can do that by actually releasing a hormone called glycagon, glycogen, I never could say that, I apologise for my bad pronunciation, but glucagon, oh, I'm just murdering that word, I'm sure, but that actually causes glucogen to be converted to glucose, and I know we're using a lot of glue kind of words here and it gets confusing, so I really recommend you download these so you can see them and don't have to just worry about my pronunciation of them, but that causes the levels of glucose to go back up to where they should be. And that ties into what a negative feedback loop is. Okay, so a negative feedback loop would be where levels get too high, the body responds to bring them back down. Levels get too low, body responds to bring them back up. That's what a negative feedback loop is talking about. The body has a set level it wants things to be at. If they get too high, it will respond. If they get too low, it will respond. That's what a negative feedback loop is. Nothing to be worried about, just something that can happen. Okay? So, levels get too high, insulin secreted, conversion of glucose to glycogen, and it's stored. Okay? Levels get too low, hormone is secreted, and glycogen is converted back to glucose, the levels rise back up. Okay? So, that's controlling blood sugar. Okay? You've probably heard of diabetes. Diabetes is a condition where blood sugar levels go high and they remain high. This is usually due to the body not responding to insulin or insulin not being released by the pancreas, depending on the type of diabetes you've got. There's actually type 1 and type 2. Type 1 is an inherited condition, it's genetics, and we'll come on to genetics and how we look at genetics in a bit. So it's inherited, it's passed down through generations, that's type 1. And type 2 is actually linked to diet and obesity. So people who maybe have a poor diet might be more likely to develop type 2 diabetes in the future. There is a link between that, between obesity and diet to type 2. So type 1 is inherited, type 2 is developed by someone based on their diet and their lifestyle. With these levels of glucose go high, it can lead to shock. Similarly, the levels can drop too low as well. They can be controlled by insulin injection, so insulin can be injected, and in fact in terms of type 1, that's really the only way to control it, by injecting insulin, artificial insulin, which can be produced, and we actually go on to see how that can be done, to bring those levels back down. So when glucose levels get too high, people who suffer from diabetes generally have to monitor their blood sugar levels using a device, take blood samples or a fixed device, and then they have to inject insulin if the levels are too high to bring the levels back down to where they should normally be. So it's the same sort of process as before, where blood sugar levels need to be controlled, but with diabetes they have to be controlled artificially by insulin injections. Whereas type 2 we can possibly prevent if we improve people's diet, and that's something that is obviously important to all of us and something that they're looking at nationwide in terms of improving people's diet and lowering the number of cases of maybe type 2 diabetes and other obesity related problems. Another one, now this one is one that comes up mainly, well I think only in the higher paper I should say, is talking about the thyroid gland. The thyroid gland is responsible for producing the hormone thyroxine. Can say that one, fortunately enough, and that controls the metabolic rate. The metabolic rate is how fast your body respires and breaks things down, and chemical reactions happen within the body. This uses a negative feedback loop as well, and this diagram sort of shows you how this process actually works. Again, it's monitored by the hypothalamus. If the thyroxine levels are too low, what it will do is it will release a hormone which is TRH, thyroxine releasing hormone, which goes to the pituitary gland, which releases another hormone called thyroxine stimulating hormone, which is sent to the thyroid gland, which is like sort of in the neck, and then that causes the thyroid gland to release thyroxine into the bloodstream. If the levels rise up, then the negative feedback is that's picked up back again by the pituitary gland, and it stops producing TSH or thyroid stimulating hormone. So the process then stops. It doesn't need any more thyroxine in the blood, so it feeds back and it stops producing it. If the levels are too low, then that will be picked up by the hypothalamus, and that will cause that process to happen again. So again, we've got our levels here. If the levels drop too low, the body responds and brings it back up. If the levels get too high, the body responds and brings it back down. Negative feedback loop. Specific thyroid gland for thyroxine. And also adrenaline is not on a negative feedback loop, but is one of the ones in the higher. You have to know that if the body's in danger, it will release levels of adrenaline, or adrenaline will go up in the bloodstream by the adrenal glands, and then those will cause the fight or flight response in the body. But thyroid gland is one that's in the higher paper, and one you need to know about. And another good example of a negative feedback loop. Whereas adrenaline isn't on a feedback loop. Levels go high and then they just fall, because normally you don't have very high levels of adrenaline in the body, it's only when needed. Whereas this, you should have levels set at a certain amount at all times, really. It can lead to, they could ask you, if levels of thyroxine or a condition that people suffer from is underactive or sometimes overactive thyroid. Underactive thyroid can lead you fatigued or lethargic and tired. It can also impact on growth as well, can cause stunted growth. So that's why it has to be sort of looked at if people suffer from an underactive thyroid, and an overactive thyroid would speed it up too much, and could also be something that we wouldn't want. So hormone control of the menstrual cycle is an important one, or hormone control in fertility. As I said, you need to know about the testes, and they produce testosterone, which controls changes during puberty, and the ovaries, which release estrogen, which controls the changes during puberty for a female. But also you need to know a little bit more about the menstrual cycle. Now the menstrual cycle is a cycle of eggs within the ovaries maturing and being released every 28 days. Slightly different in males and females, and this is just sort of more an interesting fact, I doubt it will come up, that with the eggs within the ovaries, essentially when a girl goes through puberty, she creates all the eggs she needs for her entire lifespan during that time, and then she doesn't produce any more sort of later on in her life. It's during puberty you develop all the eggs you need, and then they just sit in the ovaries or wait in the ovaries in an immature state, and then this process actually causes them to mature and then be released. Whereas in the male, testosterone causes sperm to be produced constantly almost through life. So it goes on longer. And women, obviously, when they get to a certain age, will go through the menopause, and the menstrual cycle will actually stop happening after that point. So this is a cycle, and the length is about 28 days. They can ask you about that. Sort of once a month we generally sort of think, but the cycle is actually 28 days. That's the one they need you to know or recognise for GCSE. It's controlled by essentially 4 different hormones, and you need to know the names of them, the abbreviations of some of them, where they're produced, and what their role is in this cycle. Quite often they'll give you a graph which shows levels of these going up, and they show 2 things. They show the point of ovulation in the cycle. That ovulation is when an egg is released. And they also show the thickness of the uterus, which is the womb, essentially, and how thick that gets with a blood-rich lining to be able to support a fertilised egg if it was to embed in the walls of the uterus, or if the egg was to be fertilised, it could keep it alive while it starts to grow. So often you'll see a graph that shows the patterns of these. I haven't put one on here, but if you go to BBC Bite Size as a website, they've got a good example of that. These are the hormones that are actually controlled, controlling the process of the menstrual cycle though. You've got FSH, which is follicle stimulating hormone, and that's the sort of first one in the process. What that does is that's produced by the pituitary gland, and it actually has to be sent down to the ovaries, and it causes an egg to start to mature. So those eggs that I said that were present in the ovaries, one of them starts to mature. When non-identical twins occur, that's where 2 eggs have actually been stimulated to be matured and then released at the same time, and that leads to non-identical twins. But ideally, 1 egg is stimulated to mature and actually gets released. The important bit about the menstrual cycle is the hormones that are involved in it are linked together. So generally, when one's released, it stimulates another one to start to be released, and the process moves on to the next stage. With follicle stimulated hormone, FSH, it stimulates the ovaries themselves to start releasing estrogen into the blood. Estrogen, produced by the ovaries, actually stops FSH being released by the pituitary gland. It actually tells the pituitary gland to stop producing FSH. It moves on to the next stage. And what it does is it actually causes the lining of the uterus to thicken and get ready to receive a fertilised egg. As I said, it then passes on to the next stage, because what it does is it actually tells the pituitary gland to start producing another hormone called LH, or luteinizing hormone. That starts to be produced or released by the pituitary gland. So again, it takes a bit of time. Hormone responses are generally a bit slower, and this whole process takes 28 days. But it causes the next stage to kick in, luteinizing hormone, to be released by the pituitary gland. That causes that egg to actually be released. So the egg's matured because of the follicle stimulating hormone. It's caused the egg to mature, and the uterus lining's been thickened by the estrogen, ready to receive the egg, and then luteinizing hormone causes that egg to be released. So the egg actually gets released, and it starts to move on to the next stage where progesterone can be involved. Now, progesterone actually maintains the lining of the uterus, makes it thick and keeps it thick in the middle part of the menstrual cycle, so that if that egg is actually fertilised and makes its way down the fallopian tubes to the uterus, it can embed in the lining and actually start to grow and develop. Progesterone levels stay high during pregnancy because obviously you need to keep the uterus lining thick during that time. But if the egg is not fertilised, then progesterone levels drop back down, the uterus lining starts to recede and break away, and that's when it leads to a period, the last part of the menstrual cycle where the blood is released during this process. Obviously, once this has happened, the cycle then starts again, and a new egg is caused to mature, the uterus becomes ready, the egg is released, and the cycle carries on and moves on around again. Okay? But those are the four hormones you need to recognise what they do, where they're produced, and what they then inhibit or pass on to the next stage. You can remember the ones FSH and LH, the ones that end in H, they're the ones produced by the pituitary gland, and the other two, estrogen, which begins with an O, is produced by the ovaries, and so is progesterone. Okay? So those are the ones you need to recognise and know what they do. Contraception is another thing related to those hormones that we've talked about, and hence it's tied into this topic to do with fertilisation. You get hormonal and non-hormonal methods of contraception. Hormonal methods, we're normally talking about the pill. We can be talking about the implant, which can release hormones into the bloodstream over a longer period because you don't have to keep taking it. It can be just left in there to do what it needs to do. But generally, if you're thinking of hormonal methods, you're probably thinking of the pill. They usually contain estrogen and sometimes progesterone as well, sometimes just progesterone. They varied it over the years. Early pills used to contain a lot more estrogen than they do now. There's certain issues with that, so they've sort of changed it. It's got more progesterone. Essentially, what I like to say it does is it fools the body into thinking that it's pregnant. When the body thinks it's pregnant, it clearly does not want to release another egg, so it makes the body think that. And then when you stop taking it for a short period, the body actually realises what's going on, the uterus lining can break down and it passes on to the next stage. But before it actually starts to stimulate and develop the release of another egg, you start taking the pill again, keep the progesterone levels high and stop the process. Because the levels of progesterone are high, it actually inhibits the production of FSH, follicle stimulating hormone. So therefore the egg inside the ovary never actually matures and never actually gets released. Good things about the pill or hormonal methods are they are about 99% effective. They can be affected if you're taking the pill, particularly if you have diarrhea or vomiting. That can cause you to not actually get the required dose of hormones and can cause this process to be interrupted, but they're 99% effective. In most cases, if you're on the pill, you will not get pregnant. So that's a good thing to them, because that's why you would be taking it. The cons are there are side effects. They can cause mood swings, they can cause weight gain, they can cause a few problems like this, and that's the downside to taking them. particularly for long periods. Non-hormonal methods, now there's differences to each one. I mean, there's barriers such as a condom, which actually is a barrier to prevent sperm reaching the egg. That could be the diaphragm as well. It prevents sperm reaching the egg. It forms a barrier to stop that process happening. There's IUD, intra-urethral device, which is essentially an implant which prevents the lining of the uterus from thickening and therefore cannot support a fertilised egg. There's spermicidal gels, which can be used to kill the sperm and can stop fertilisation in that way. Some of the problems with some of these, though, is they can be less effective than this method because they can go wrong. Sometimes not all the sperm are killed, for example. Sometimes the condom can break and cause problems to occur in that regard. Surgery is another non-hormonal method, vasectomies and hysterectomies, but these methods, although effective, are often permanent and therefore are not something that have to be considered before actually undergoing surgery to prevent conception, which is having a baby. So this is a method of contraception. So these are, I mean, often questions will ask you to compare I'll start again. Hormonal, it's a Friday evening, I apologise. Hormonal and non-hormonal methods. So I'll ask you to compare one and what they do and maybe give an example or talk about the pros and cons of each. I haven't listed the pros and cons of these because they're all slightly different. The pill or hormonal control is very effective, but there are some of those side effects to them. So that's like nervous response and hormonal response. That's that unit, one of the big units in B2. We're moving on now to sort of genetics and inheritance and that sort of side of things, which is the second topic, if you like. So we need to know about genes, which are small sections of DNA. You need to know about DNA, which is deoxyribonucleic acid. That's a long strand which, when coiled up, forms a chromosome, and then chromosomes are found inside the nucleus of a cell. As I say, generally you're talking about humans when you're talking about inheritance, not always though, talking about chromosomes. You need to be able to recognise that humans have 46 chromosomes in 23 pairs. They pair up. Just one set of those, or one pair, is responsible for gender, and we're going to look at the control of gender in a little bit. And the other 22 pairs are responsible for all characteristics you have. All the physiology of the body are controlled by these other chromosomes. So essentially you need to know that breakdown. Cell, inside the cell is a nucleus. Inside the nucleus you find chromosomes. Chromosomes are coiled bits of DNA, which is essentially like a long strand, which is a double helix shape, made up of like a phosphate backbone and base pairings, which they can ask you a bit of detail about. And then a small section of that DNA, if we just took a small section of it, that would be a gene. And what a gene does is a gene, when it's in a pair, actually contributes to a characteristic. So sometimes some characteristics are controlled by only one gene, sometimes it can be more than one gene, and the combinations of them can lead to it. Essentially it's a code that causes a building process to build up proteins within the body and actually cause the development of certain characteristics. It can have problems. There can be defects in the genetic code or in the chromosomes which cause problems to occur, and we look at some genetic disorders a bit later on. Two types of cell division you need to recognise. We talked about mitosis. I'm going to talk about meiosis in a bit more detail. I did a diagram of mitosis last time. You actually got asked a cell cycle question about what goes on when mitosis is happening in an embryonic stem cell. Meiosis is the other type of cell division. Mitosis made my toes, it made every cell in my body in fact, it's used for growth and repair, whereas meiosis made me. Meiosis makes the gametes, which is the name for the sex cells, the sperm and the egg that actually need to combine in fertilisation to actually produce a new organism. So, mitosis makes all the cells in the body, it's used for growth and repair, it only divides once and it makes 2 new identical cells. Not going to say any more about it because that one should have been in B1, but it's worth bearing in mind because they can ask you a bit about it because you should have done B1 already before doing B2, so be aware of it. Meiosis, on the other hand, is used to make the gametes. There's actually 2 divisions that take place and it ends up making 4 cells at the end which are non-identical. They're not identical to the parent cell. The parent cell is called diploid, which means it's got 2 pairs of chromosomes for every chromosome that it's got in it. They're in pairs. The daughter cells in this are called haploid, which means they've only got half the number of chromosomes needed for a full organism. Because we're talking about the sperm and the egg. We need the sperm and the egg to have half each because during fertilisation they fuse together and that creates a diploid cell which has got the full amount of chromosomes. And I'll hopefully show that on a diagram here I'm going to do for meiosis. So for meiosis you start with your parent cell. Again, I'm going to draw the cell without the nucleus, or maybe it's just the nucleus, but I'm not going to put it in it, but I'm going to show some of the chromosomes that are present there. So that's our cell or our nucleus, and we've got some chromosomes in there paired up. So we've got one like that, and one little one, and another one. Now, as I say, if this was human, if we talk about human, there would be 46 chromosomes in 23 pairs, so they'd be paired up in 23 pairs. And this cell would be, they would pair up at the start of the process. If it was female, this would be the ovaries. If it was male, it would be the testes. That's where the gametes, the sex cells, are produced. Anyway, those chromosomes actually are paired up like that, and then cell division starts to take place when they sort of line up. But the first thing that needs to happen is it actually needs to duplicate itself. It needs to create copies. So it actually copies the DNA or the chromosomes first of all, so there's actually twice the amount. So as I say, if this was a human cell, there's 46 chromosomes at this point. And the first thing is we're going to double them up. We're going to create copies of them, so we've got twice the amount between them. Now, I'm not actually going to draw my diagram, but it's at this point when something very interesting happens, and it's important because it leads to variation. You would be a mix of your parents' genetic code. So you'll have characteristics that are probably similar to both parents because you're a mixture. Half of your genetic information came from a sperm, half came from an egg. Also at this point, you're not going to be exactly like either parent, and the way it leads to variation and causes it is there's actually crossover of sections of DNA here. They actually swap little segments and they essentially just jumble up the genetic code, and this leads to the variation that's produced. Anyway, the cells divide at this point to produce 2 different cells which have the full amount of chromosomes at this point. So we've got one of each in each. Now they wouldn't be exactly the same because they would have swapped bits, but I'm not actually showing that in this diagram. But the important bit here is at this point, rather than duplicating anything, the cells actually divide a second time. There are 2 divisions in meiosis. So the cells have divided once. Duplication occurs here. It makes a copy of all the chromosomes. They swap over little sections, and then it divides. And then it divides again straight away afterwards. And it produces 4 cells rather than 2. So this is where my diagram using colours isn't very good. Because of that swapping that went on, each of these chromosomes would be slightly different to each other. So when they divided to make this, I mean, if you look on my diagram, it looks like these 2 are the same and these 2 are the same. That would not be the case because of that crossover of bits of DNA. Each of those cells would be different to each other. Each one is slightly different. These are the gametes. I'm going to make them sperm cells. This is going to be a male, so I'm going to put a little tail on them. There we go, we can clearly see their sperm cells now because of the little tail. These have 23 chromosomes rather than the full 46. Because the other half is going to come from, in this case, the mother in the egg cell. She's going to provide the other 23. So this is just one set. These are not paired up like they were in the parent cell, which would be, if this was the male, this would be the testes to produce sperm. It could similarly be the ovaries to produce eggs. But these are non-identical cells. They're not the same to each other, and they're not the same to the parent cell that produced them. 4 different daughter cells are produced. That's what's important about meiosis. So the reason meiosis is important and the variation it creates, it's important for sexual reproduction. Sexual reproduction is the joining of a sperm and an egg. Or, I say sperm and an egg, that's bad because I'm only really talking about animals in that regard. It could be the pollen and the ova or ovum in a flower. So it's really the joining of 2 sex cells combining together to produce a fertilised egg, which can then go on to divide to become an embryo, to become a new species, or sorry, not new species, that's bad, a new offspring of the parent. The good thing about sexual reproduction, because it's the fusing of one of these gametes with another gamete, it leads to variation. All the offspring are different to each other. So I said, you are half the genetic material from your father and half from your mother, and that's created you. But you're also not identical to any brothers or sisters you may have. And that's because of this sort of mixing of DNA and the fact that all these cells are different. Every time a sperm cell fertilises an egg, a different combination is created, which will not be identical to the previous one. So it leads to this variation in sexual reproduction, which is great for many reasons. It allows us to evolve, and it allows us to all be different, which is interesting, but also allows us to not maybe be susceptible to the same things. We all have slightly different, say, immune systems. One thing that is a defect in one of us is not a defect in all of us. So these are good things about sexual reproduction. Some species can do asexual reproduction. Asexual reproduction involves only one parent. It can just be a cell dividing through mitosis, so bacterial cells will be able to divide and recreate themselves on their own through asexual reproduction. They just create copies of themselves and can cause that to just keep going on. And that's quite good because it can happen very quickly for bacterial cells. It can happen sort of once every 20 minutes, they can sort of reproduce themselves. But it does result in identical copies, clones, which can be bad because one thing that affects actually one of them can actually affect all of them. A good thing is a lot of things that can do asexual reproduction with only one parent are capable of doing sexual reproduction as well, which allows them to vary themselves and therefore evolve and change over time. Key things though, asexual reproduction is just one parent. A flower, or a plant, sorry, can reproduce itself. The pollen from the same flower can reach the egg of that same flower, but it can also be passed on via bees and other insects, pollinators, to other flowers and fertilise them as well. So it can do both sides of reproduction. This is good because it only needs one parent and it can happen quite quickly, whereas this is good because it leads to variation within a species, but does need 2 parents to actually occur. So we talked about sexual reproduction in terms of inheritance. So the characteristics that you have are controlled by genes, and those genes are small sections of DNA responsible for your characteristic. The single form of a gene is called an allele. So as I said, your chromosomes are in pairs and each of them will have a gene on it. And if you've got just one form of that gene, it's called an allele. It's just a single allele. And the combination of those 2 alleles that you've got on your chromosomes actually gives you a different characteristic. These characteristics are called your phenotype. That's the characteristic you might have. So phenotype, and the way you sort of remember that, or I try to remember phenotype, is this is your physical characteristics. Okay, So that's what you're physically like. That for me would be my sort of dark brown hair, my hazel eyes. Those characteristics are my phenotype that were caused by my genotype, which is the combination of these alleles actually giving me these different characteristics. So that's your genotype. And again, you can remember it because it's G, but that's your genetic information. Okay? So the information that causes you to have those physical characteristics, that phenotype. When we're talking about genes, often they're represented with letters for different alleles. Now they use a big letter for a dominant allele. A dominant allele means if you have any form of that allele in your genotype, that will mean you have that characteristic. An example would be dimples. Dimples is a dominant allele, which means if you have any of the gene for dimples, dimples are like when you smile, you get those little indents in your face. This is a dominant characteristic, so if you have any of the alleles for dimples, you will have dimples, regardless of whether you've got it on one allele or both alleles. And I'll show you the combinations that I mean in a second. Whereas when we're talking about recessive alleles, they're often given small letters. These are ones that you only have that characteristic if you have two of those alleles in the same gene, on the same chromosome. So, for example, you could have this genotype. This would be the genotype, and I'm using big letters here to show you have two of the dominant alleles. So this would be the two alleles of the gene on the small section of DNA. These are both the dominant gene, and therefore you would have that dominant characteristic. That might be dimples in this case. This is another genotype you could have, another combination. You could have one of the dominant and one of the recessive. In this case, you would have the dominant characteristic, because the dominant always takes preference if you have any of the genes for it. So I would have the dominant dimples in this genotype, and the phenotype would be to have dimples. Only would you have no dimples if you've got two of the recessive alleles, two of the small letters. So if you have two of the small alleles, then you will have the recessive characteristic or the recessive phenotype. In the example I'm using, this would be no dimples. This person would have no dimples. So if we had three people with these genotypes, this person would have dimples, this person would have dimples, and this person would have no dimples because they have two of the recessive. This person could be considered a carrier for no dimples because they carry the allele for it, or the gene for it, but they don't have that characteristic. A carrier is where you carry the gene for a certain characteristic or phenotype. Can be a genetic condition such as cystic fibrosis. You can be a carrier for it, but you don't have the actual characteristic yourself. So in terms of cystic fibrosis, this would be a person who was healthy, did not have cystic fibrosis. Cystic fibrosis is a disease where you actually produce a thick, sticky mucus which can block up your airways and block up your digestive system, or where enzymes are produced. It is a serious problem for people who suffer from it. This person would be a carrier but not actually have cystic fibrosis, whereas this unfortunate person, because they've got two of the recessive, would have cystic fibrosis. Now we can use things called genetic cross diagrams to actually show the percentage chance of having a certain characteristic. Based on the genotype, we can predict what phenotypes the offspring of that person is likely to have. This is a classic example for gender determination. You need to know that the mother's genotype in terms of gender, as I said, is only controlled by two chromosomes, or one pair of chromosomes. And the chromosomes, they actually say for the mother is XX. It means those chromosomes are the same. They're both X chromosomes. That means you are female if you have two X chromosomes. Whereas the father has an X and a Y chromosome. Okay, that's if you have an X and Y chromosome, you are male gender. Now, in a genetic cross diagram, what you do is you draw the mother at the top and the father at the side. You could draw them the other way around. It wouldn't be massively important as long as you have one parent one side and one the other. And then you do the combinations that you can have in terms of the offspring. Essentially, you take the first allele from the mother and put it in this box. And then you take the first allele from the father and put it in this box. And in this case, this would be XX, X and X, XX. Then we can go over here and do the same for the second allele for the mother and the first allele for the father. We again get XX. Now if we do it again for the bottom row, we take the first allele from the mother and the second from the father, we get XY. And the second from the mother and the second from the father, we get XY. This is the combination that will be for every determination of gender, because every mother is XX and every father is XY. This shows us that there is a 50% chance of having a girl, XX, or a boy, XY. It is a 50% chance, or a 1-to-1 ratio, or the probability is a half. You could say it in many ways, but you need to say that in this case it's 50-50. Half are female, half are male. That's why the population balance is maintained on the planet. Okay? Half the children born are male, half the children born are female, and that always has to happen in any genetic cross of two offspring. We can do it for genetic disorders, and I'll do one just ever so quickly for that. I am conscious we've spent a lot of time on genetics and nervous response. We're about 50 minutes in, but they are big topics, so they're worth going over. If we were doing cystic fibrosis, so cystic fibrosis, as I said, is I don't know if I could spell for the moment. That should be enough. So cystic fibrosis, we're going to use, again we'll use letters. Now we could use C's, and quite often they use C's for the genetic cross diagrams, but C's not a good one because a big C and a little c look fairly similar, but we will try to do it. But what I'm going to do just to highlight it in my example is I'm going to use two colours for the dominant and the recessive alleles to try and show it a bit better. So a genetic cross diagram, we might be asked if two parents have a certain characteristic. So let's say one suffers from cystic fibrosis. Now if someone suffers from a recessive phenotype or a recessive characteristic, we know their genotype, and they may do that in an exam. Not tell you the genotype of the parent, but they tell you they've got a recessive disease, in which case you know they have to have two of the recessive alleles. So for the mother, which we'll put at the top, we'll say she suffers from cystic fibrosis, unfortunately. So her genotype has got to be two of the recessive little c's. Whereas the father, who's on the side, we'll say he does not suffer from cystic fibrosis, but he is he's a carrier. So he has one of the dominant alleles and one of the recessive. He's a carrier. He carries it, but he does not suffer from it himself. Now we draw our genetic cross diagram. You can do it very simply, quickly in an exam paper, something like that. And we'll just put it down there, and then we will do the combinations that we get in terms of the father and the mother. It looks like, I don't know, we might have some glare on the screen causing some problems at the moment. It's always a problem because we always do these in the summer because the exams are in the summer, and obviously the sun comes out, and yeah, that's great because we get to enjoy it, but the live stream doesn't work quite as well. Hopefully you can still see what I'm doing on the board, and you can certainly still hear me talking about what we're doing. So anyway, when we do the genetic cross diagram, we do the father's first chromosome, and we'll put it in here, and we'll do the mother's chromosome, first chromosome, and we'll put it here. That's the first combination. Now, we'll do the same for the father's first and the mother's second. It doesn't matter what order you do these, but you just fill them in. Then we'll do the mother's and the father's there, and the second and second there. Now what we do is we look at the combinations that we've actually got based on the offspring. These are the chance of the offspring being born. It doesn't mean if these two people had 4 children, they would be these 4 combinations. It means every time they have a child, there is this percentage chance that their offspring will be these genotypes and therefore will have those characteristics. Now, the question might be, how many of the offspring, or what is the percentage chance of the offspring of these two parents having cystic fibrosis? In which case, I would look at the ones that do. They need to have 2 of the recessive. These are the 2 that do. So there is a 50% chance that they have or suffer with cystic fibrosis. Okay, 50% chance that they would suffer. That means every time these two people have a child, there's a 50% chance that they will suffer from cystic fibrosis. And this other combination that we can see here, there is 50% chance they are a carrier, because those other two offspring carry it but do not suffer from it. Okay. Now there was a question that came in, I've just been shown, that there's some key words. Homozygous and heterozygous. These are talking about the genotypes that you can encounter. So with homozygous, what that means is the two alleles that are present are the same. So in terms of homozygous, homo means the same. Okay? Homozygous means that the two alleles present are the same. And there's actually two combinations for that. There's two of the dominant, the big C, or the little c. Okay, they would both be homozygous. And then there's heterozygous. Hetero means different. Okay, so for heterozygous, what you actually have is each allele is different. Which there's only one combination that could be, and that's with the big and the little. In this case, it's a carrier. So there's some key words, and sometimes in exam questions, they just refer to the parents as one parent is homozygous and doesn't suffer from cystic fibrosis, in which case you know there'll be two of the big ones, or they're homozygous and they suffer, in which case you know there'll be two of the little, or they'll just say one of the parents, the mother, is heterozygous, and they won't tell you, but you need to recognise that heterozygous means they have one of each of the alleles in their genotype. So that's a genetic cross diagram. Now you could use genetic cross and what's called a pedigree analysis, or like a family tree, to try and identify the different genotypes of the people in it and whether they have a certain phenotype or characteristic. I'm not going to go through everything in it, but you have different generations. This is the first generation, second generation, third generation and fourth generation in this family tree. They can ask you about people in different generations. And what they're generally looking for, a few tips with it are, if someone has the recessive characteristic, you already know their genotype, so write it above them. This person, Esme, has sickle cell anemia, which is recessive, so we know, because she suffers from it, she must be homozygous with two of the recessive alleles, because that's the only way she would suffer. Similarly here with these people, all these sufferers must be homozygous with two of the recessive genes if they've got the recessive characteristic. Then, generally, if someone is homozygous with the dominant and doesn't it, all of their offspring will have the dominant phenotype. So, in this case, none of their children would suffer from sickle cell if they were purely homozygous dominant. Okay? But in this family tree, as lots of the offspring end up suffering with sickle cell anemia, the recessive characteristic, then it leads us to believe that these people, who we don't know, who have the normal cells, but they're likely to be heterozygous, have one of each, or be carriers, in which case their offspring suffer from it. If they were homozygous and dominant, none of their offspring would actually have the genetic disorder. And do a genetic cross with someone with two of the homozygous dominant, and you'll see why, if you get it correct, that none of their offspring, there's a 0% chance they will have that genetic disorder. So, variation within individuals, I said sexual reproduction is good because it leads to variation. Variation can be controlled by genetic factors, in which case the sperm and the egg and fertilisation in that regard can lead to genetic factors. And then environmental variation is where there's a change because of the environment. That could be diet, it could be something as simple as these are two twins, one dyes their hair, that's an environmental factor. Some can be controlled by both. So your body shape can be controlled by both genetic factors and environmental factors. You could be predispositioned towards a certain body type, but if you have a bad diet, you will change that. So it's something to bear in mind that some can be both. Often mutations within genetics or the genetic code will lead to variation or cause changes. But some of those mutations can be really useful, and they lead to adaptations that can actually be a positive for the species and actually cause it to survive better in a certain environment and therefore actually get on better. These mutations lead to evolution, and there's some ideas of evolution you need to be aware of. Now, there is creationism, which believes that all life was created by another being, and this is something you do actually need to be aware of, that is a theory for the development of species. But the two main ones that sort of come in are Lamarck's theory, although they don't often name Lamarck as the person who suggests it now, but in the GCSE, but they can do, so it's worth being aware. And Darwin's theory of evolution by natural selection, which I'll go into a little bit more detail on. Lamarck suggested that if an animal used a characteristic enough, they would develop that characteristic and they would actually change that characteristic, and then they would pass on that characteristic to their offspring. He claimed that we all evolved from like worms, and worms essentially did enough stuff that they actually changed their physiology to actually evolve and to change their actual makeup to become a different species. A classic example of Lamarckism is when you look at giraffes. He sort of suggested that a giraffe used to have a short neck, or the ancestor of the giraffe used to have a short neck, and just by sort of stretching its neck enough to reach leaves in trees, it actually grew, and when it grew its neck, then when it had children, its children were born with a long neck already, and hence the giraffe was actually created. That theory's been kind of debunked now, because we know it doesn't really happen. We've seen enough evidence to show that it doesn't. It would sort of suggest that if I went to the gym every day and worked out, got even buffer than I already am now, if I had children, they would inherit that characteristic and they would be born naturally like muscular because of it. That sort of theory is kind of, we've gone beyond it. We sort of look at Darwin's theory of natural selection as the successful way that evolution happens. And you have to know that process of natural selection. Now, natural selection starts with the fact that within a species there is variation. This variation is often caused by mutations in the DNA. Small changes in the DNA actually cause species to be different. Okay? So those species being different, for example, if we go back to the giraffe, what it means is the ancestors of the giraffe, the animal that the giraffe originated from, was just a short-necked animal. Because of a mutation in the DNA, one of these animals with a short neck gave birth to a child with a slightly longer neck because of a mutation in the DNA. Unfortunately for the parent though, all the food that was on the ground, other animals ate. So there was very little for it to feed on. But its offspring that had a long neck actually could reach the trees and could reach the leaves up in the trees. And because of that, it was more successful than its parent. Because it was more successful, it actually caused it to live longer. And what all animals do, if they live longer, it means they're more likely to survive and actually reproduce. The longer an animal lives, the better its chances are of finding a mate that it can reproduce with and actually have children. And because of that mutation in its DNA got passed on to its offspring, it actually passed on that advantageous characteristic, such as its long neck, to its children, and they had long necks, and they were then more successful, and they had more children, and it carries on. And over many, many generations, it can actually lead to the evolution of a new species. When it becomes a new species is when it's no longer capable of interbreeding with the original species, the ancestor, the animal with a short neck that actually it originated from. When they can no longer interbreed to produce fertile offspring, it's become a new species, and it can no longer breed with the original ancestor species. Okay? But that's a very brief, a very quick whip through natural selection and how evolution is suggested now, and that's what you need to know for your GCSE. We found out evidence for this by looking at fossil records. Fossils are the remains of an organism preserved in rock for thousands, if not millions of years. We've got pretty good fossil records going back to ancient times. We haven't got complete fossil records for very good reasons. Fossils can be destroyed, they're encased in rock and they can be easily destroyed by like volcanic activity, by erosion and weathering of rocks, things like that. They can also, some animals do not have things that can be fossilised. A jellyfish is a good example. There's very little material in a jellyfish that can actually be preserved in rock and fossilised, so we may not have fossil records that show us some of the ancestors in the past. So there are gaps in our fossil records, but generally we've got a good picture going back millions of years to like dinosaurs and things like that, and even before of prehistoric animals that we can base these ideas of evolution on. There's a classic one in the textbook, and you can see it on like sort of websites of fossil records of the ancestors of the horse. They showed that the horse used to have shorter, stumpy legs, or I say a horse, it was an ancestor of a horse, wasn't the horse itself. Had short, stumpy legs with wide toes because it lived in swampland. But once the swampland started to dry up, those short, stumpy legs weren't as good, and it would get caught by predators that were preying on it. So all the short, stumpy legged little ancestor horses died out, and the offspring had longer, more sort of stronger legs with just one hoof, could actually run faster on the ground, and they survived, they had offspring, and eventually they evolved. And we've seen fossils showing that change over time. Fossils can also give us evidence not just of evolution, but also of extinction events. Extinction could be caused by a new predator moving to an area, causing something to be upset. A new disease being introduced to an area and causing the animals to die out. It can be caused by loss of habitat, and this ties into some of the later topics, which is about humans destroying ecosystems and destroying habitats, causing a reduction in biodiversity, and can actually cause habitats and species to die out. It can be caused by a more successful competitor arriving. So sometimes we introduce, unwittingly, humans introduce a more successful competitor. For example, with that, you could talk about red squirrels and grey squirrels. Grey squirrels got introduced to the UK. They're more successful than red squirrels. They're more aggressive. They have a more varied diet, and they've actually taken over a lot of the areas where red squirrels used to live because they were better at competing. They're not a predator. They don't eat red squirrels, although they are more aggressive and they can kill them if they come into contact. But they don't eat them. They didn't necessarily bring a disease, but they are better at actually surviving. It can be caused by rapid climate change, and climate change and global warming, something we look at. It can be caused by a mass extinction, can be caused by a big catastrophic event such as a meteor strike, and they say that's a theory for the extinction of dinosaurs, or a volcano eruption could cause a mass extinction. There's reasons like that, but these tend to be the ones that there's more evidence for extinctions based on these different sort of factors. To show evolution, you can use something called the tree of life. Now this is quickly up here. This is how we can classify different organisms. Kingdom, phylum, class, order, family, genus, species. It's something that you need to remember, that sort of order. You can be asked to, sometimes they'll give you this with some of them missing, and you have to pick them out and fill them in the correct order. You could be expected to remember the whole order. I've got an acronym for remembering it that I learned at school, but the one I will share with you, because I can't share that one unfortunately, is kids prefer candy over fresh green salad, is one you can use. But kingdom, phylum, class, order, family, genus, species. It's how we classify organisms into little groups. So some of the kingdoms we have quite in common with lots of other animals, but when we get down to the genus and species, Homo sapien, we're a bit different. And some of these are like all given weird Greek names, some of them quite funny, like the brown rat is Rattus ratus. And you get like Canis lupus is a wolf, Canis familiaris is a dog, which are very similar. So the Canis is the same, the genus, but the species, familiaris or lupus, are different, and that means they're different species and can't breed with each other. You can't breed a wolf and a dog anymore, although they're related, they're no longer capable of interbreeding. All breeds of dog, though, are the same, so they can interbreed, and we come to that in a second. Anyway, we can see that on a tree of life or an evolutionary tree. What this shows a common ancestor in all these species, A through to G. All of them had a common ancestor in their past. And when the tree branches, that's when the species start to divide. So they can ask you things about what's the most common ancestor between species F and G. Well, if they were asking about F and G, the most common ancestor to both of them, we'd follow it back where they meet to where it meets another one. So that would be species E. It's divided there. So you can go through and you can look at it like this. Sometimes you'll have species at different levels and the tree will branch off. This is just one version of it. They can show you it sort of multiple different ways or lots of different ways, because this is where the species actually divided and became separate species, and maybe a species went extinct. This is an ancestor species sort of down here. So you've got that where you can see commonly related species to each other. As I say, it doesn't have to be in that format. They could give you it in another format. You can see different structures of this, but it's about looking where those divisions happen and looking about, this would be the ancestor they all had in common, this point here, and then they've divided up over time. So the closest ancestor of G is actually F, because it divided at the latest point on this family tree, as we can see. Animals and plants and things are not the only things that can evolve over time. Bacteria can evolve, and this is a problem for us because they're actually evolving to become resistant to antibiotics. Antibiotics are the drug used to kill bacteria. Antibiotic resistance is actually built up through a process of evolution, but because bacteria recreate themselves or reproduce so quickly, it can happen relatively fast. Evolution usually takes thousands of years to happen. Bacteria can do it a bit quicker than that because of the speed in which they reproduce. And what it is you start off, when you're infected with an infection, a bacterial infection, you'll have all of those bacteria will be slightly different to each other, just like the variation within species, bacteria will be varied. Some of those bacteria will be stronger and some will be weaker than others. When you take antibiotics, the weak bacteria will be killed first of all because they're the least resistant to it, so they'll be wiped out very quickly. If you don't complete the course of antibiotics, you think, well, I've killed all the weak bacteria, I'm not feeling unwell anymore, I feel much better, so I'll stop taking the antibiotics. Then you actually can get another infection where the strong bacteria that are left, that haven't died, actually reproduce themselves, and then they come back much stronger. If this process happens over time, the bacteria can eventually become completely resistant to an antibiotic. And this is a worry with some bacteria. They're resistant to lots of different types of antibiotic, and they do anticipate that people who get infected with this, there could be sort of 10 million deaths by the year 2050 if we don't find another method of killing these bacteria that antibiotics no longer work on. So they evolve by killing the weak ones, the strong survive, the strong reproduce, and they pass on that strength, that characteristic to their offspring, and have now become resistant. Humans throughout time have tried to guide evolution in the way that they want. There are certain characteristics, because of the variation within species, that humans want. It might be a plant that grows particularly a lot of fruit, or grows particularly big, or is particularly resistant to predators or pests that eat it, insects that may eat it. And if we want that plant, we'll breed that plant deliberately, we'll pick that plant and we'll breed it with another version. So, for example here, we grow a set of plants, this is population 1, the first sort of level, and we take these two plants that have grown the biggest, because we think they're the biggest, they're the strongest, so let's breed those two together, and population 2 are still taller, taller than the first generation. Now there are some that are not as tall as all the others. We would then do the process again, so we would take the two biggest from that and we'd breed them together, and we'd have the third population, which should be bigger still. We keep doing that to breed in these characteristics that we want. In this case, the size of the plant that grows, we want it to be bigger and bigger, so we're breeding together the biggest plants with each other in the hope that their offspring will inherit that characteristic and also be big. We'll still see variation, but the general trend will be an increase in size. That's what happened with dogs. So dogs were related to wolves, but we bred wolves with the characteristics that we liked. and we kept breeding them until we developed sort of new species, which was the domesticated dog. And then even within that, we've bred species of dogs together to create dogs with certain characteristics that we like. And this can cause problems. Crufts comes under a lot of flack because they look for certain characteristics in certain breeds of dog. For example, in a pug, if you know pugs, they're the ones with the sort of big eyes that bulge out the front, little dogs. They like that characteristic, the eyes that are big and bulging. That's a good characteristic in a pug, supposedly. So they take the pugs with the most bulging eyes, and they breed them with the other, the male and female pugs with the most bulging eyes, and they end up with offspring with even more bulging eyes than the parents had. And that can lead to problems, because now those dogs have problems with their eyes, can have difficulty later in life due to eye problems, subject to like cataracts and things like that. So it's actually caused problems within that breed. And that's just one example. There can be lots of others because of this selective breeding. Because selective breeding is choosing organisms with the characteristic that you want, breeding them together, and then doing the same over many generations to try and change the characteristics in the offspring. Genetic modification is probably, I think, one of the harder topics in this unit. I'm going to go over a brief overview for it, but we are getting close to time, and there's still a few things I want to cover, so I might skip ahead through some of them. But genetic modification, I'll quickly go over. Genetic modification is where you actually alter the DNA of an organism. Now that can be a plant. If a plant's highly resistant to a certain pesticide, you might spray on a crop, and you want it to grow well, you might cut a gene out of an organism that's resistant to it already and insert that into the plant that you want. And then you grow lots of copies of that plant by asexual reproduction, by taking cuttings or by fertilising the plant itself. And then you've got a whole species of plant that are resistant to a certain pesticide. So you can do it by cutting sections of DNA out, using enzymes and inserting them into the genetic code of a new organism. But another example, I said we'd come to this, is how we produce insulin for people that are diabetic. We actually take the gene for insulin production from a healthy individual, and we cut that gene out using enzymes from their genetic code. We've now mapped the human genome, we've mapped all of the genes of a human, and we've identified most of the genes and what they're responsible for in humans. We know the one that's responsible for producing insulin. So we cut that gene out, and we take a bacteria. Now bacteria are special because they don't have a nucleus. Their DNA is just floating around inside in their cells, in their cytoplasm. But they also have these circular bits of DNA which are called plasmids. And we can actually cut one of those circular bits of DNA open, and we can insert the gene that we want into that plasmid. We then reinsert the plasmid, the bit of DNA, into the bacteria, and then we cultivate the bacteria. As I said, bacteria divide quite quickly, so we cause them to divide lots and lots of times. And so we've got lots of copies of this bacteria that has the gene for producing insulin inside it. We've actually modified it so it's got that in it. We can then cause that bacteria, or that bacteria will produce insulin because it's got the gene for it, and then we can harvest that insulin and we can use it to treat people with diabetes. They can use that in their injections. So that's genetic modification. You cut sections of DNA out, genes that you want, certain characteristics, and you splice them, insert them into either another organism or into a bacteria, cause that bacteria to reproduce and get that gene that you want. Issues with it is we're not fully sure of the long-term effects of this in terms of whether it's a thing to do. People are worried because we're playing with nature, we're altering the DNA, which is the makeup of living organisms, and could cause problems. Going back to my crop example, what they're also worried about is because plants are very good at interbreeding with each other, fertilising each other, they're worried that some of that DNA could be taken up by a weed, a plant that you don't want to grow, and then that plant becomes more hardy and more resistant to pesticides and things, and it actually causes a weed to grow in your fields that is actually resistant to things you didn't want it to be, and becomes a major problem when it escapes into wild plants. Also, we don't know the effects of animals that maybe eat that plant. They will eat that plant, they'll take that DNA into their bodies. Could it then be taken into them and cause an effect in them? So these are issues with genetic modification, and certainly like research is being done to see the longer term effects. But it can be good because you can make insulin, you can make crops that are more hardy or grow more fruit or more substance for people, and lead to curing problems like hunger and things like that. So we are close to time, and we're moving into the last topic. The last topic has a lot of common sense sort of bits in it, and a lot of acquired knowledge from Key Stage 3 really, in terms of things like ecosystems. It takes it maybe a bit further, but it's got a lot of knowledge that I'm sure you guys have got already. And anyone who does geography also, I know, has covered this as well, so we'll have some of the background information. So an ecosystem is where there's interactions between communities of living organisms. And by community, I mean a group of organisms, at least two different organisms within an area. That could be based around sort of different ecosystems, could be based around just one tree, the ecosystem's living on one tree, or could be an entire rainforest. So ecosystems can be quite wide or quite small. You can get small little ecosystems that work. But it's to do with the relationships between the organisms within that ecosystem that this topic's about. This is a food chain, and there are many different sort of relationships shown in this food web, I should say. It all starts from grass, this food web. So grass is called a producer. It takes the sun's energy and it produces its own food. Every food chain really starts with a producer taking the sun's energy to feed into the food chain or food web. And that producer is eaten by a consumer. Now these are primary consumers. They're the first consumers in the chain. We actually have three in this. We have a mouse, a rabbit and a grasshopper. They all feed on the grass. They're the primary consumers. So they're eating the grass. Their energy is, well, the energy from the grass is transferred to them. They build up their biomass, which is their dry mass, and it's actually taken into themselves and they grow. Then we've got secondary consumers, things that eat the primary consumers. So we've got a snake and a lizard, the lizard eating the grasshopper, and the snake eating the mouse. They're eating those and feeding on those, so secondary consumers. And then we've got a tertiary consumer here at the top, or an apex predator, something that nothing else preys on, but actually preys on animals below it and consumes the animals below. The reason this food web is good because it shows the interactions between the different competition between the animals and the different sort of ways the food chain can actually go. So in this case, the grass is eaten by three different animals, but those animals, so say the grasshopper, let's look at the grasshopper which feeds on the grass, he's eaten by the lizard and by the hawk. So the hawk and the lizard are both competing for the grasshopper as food. And if numbers of the grasshopper were to fall, it would have an impact on both the lizard and the hawk. Their numbers are likely to fall. Because if there's less grasshoppers around, then they won't be able to find as much food, and their numbers are going to fall. But nature works in a balance quite often. So if the numbers of lizards and hawks fell because their food source was going, then the numbers of grasshoppers would probably be able to go back up because they weren't being eaten as much. And in which case the numbers of lizards can then follow and go back up. And these things tend to be a cycle where the predator animal rises up and actually the numbers starts to increase, and then the prey animal rises up as well and the numbers start to increase. And because the prey animal starts to increase, the predator starts to fall, the prey starts to fall again when the predator eats them, and then the numbers drop down, and it forms in a cycle. It goes up and down in this careful balance, in this careful relationship. Another thing to point out here though is the hawk itself doesn't just have one food source. The lizard in this food web only eats the grasshopper. So if the grasshopper's numbers fall, it will have a big impact on the lizard because their only food source is the grasshopper. The hawk, on the other hand, yeah, it might be affected because it does eat grasshoppers, but it also eats rabbits and mice and snakes. So the numbers of this will be less affected by a fall in the grasshopper because it has many other food sources it can rely on. So that's something to be aware of. When we look at food chains and food webs and ecosystems, we're looking at factors that can affect them. The two different factors are abiotic and biotic. Abiotic are non-living factors, so things that are not alive. And these can be temperature, you could be talking about, light intensity for plants, they need light to survive, moisture can be for plants and animals technically, soil pH, certainly for plants growing in a certain soil, they like a certain soil there, and wind, both direction and speed. Some plants do well where it's very windy, some do well where it's not so windy. So these things can have a big effect on the organisms that live in an ecosystem, but they're all non-living themselves. And oxygen and carbon dioxide levels. Generally carbon dioxide levels for plants and oxygen certainly dissolved in water for aquatic, which means water-living organisms. The biotic things are living factors. Okay, these generally talk about the availability of food within an area. Are there new pathogens or microbes that can cause disease? Are there new predators in the area that can cause it to wipe out? Is there competition between different species? Now these abiotic factors we can measure in different ways. We can measure using usually devices such as electronic devices. You can get a digital thermometer to do temperature, a light meter to do light intensity, to measure light intensity, soil pH, you can take the pH of the ground to ensure that that's correct, wind speed and direction you can measure. Whereas these oxygen and carbon dioxide levels, they're generally shown by indicator species. Some organisms are very sensitive to changes in their environment, abiotic changes. A good one for air pollution that comes up is lichen. It's spelled like lichen, but it's pronounced lichen. And they're the like mossy growth you see on trees. When you have lots of lichen around, it means the air is very unpolluted, it's very clean air, and these can thrive and grow in large numbers. When the air is polluted, you won't see as many of these. So they're called an indicator species, something that's sensitive to changes in the ecosystem or the environment. Water pollution, they talk about aquatic invertebrate. And depending on the levels of pollution within the water, you can get different organisms that are present. If the water's very clean, you get mayfly larvae. Whereas if it's very highly polluted, you get things like called sludge worms and stuff like that just sound horrible. Often with exam questions on that, they'll give you a chart with the water pollution levels and the different organisms that are present, and then they'll ask you to talk about different reasons why the water might be polluted and what the levels of organisms in different waters they tested are actually telling us. Generally, the more polluted a water gets, the less oxygen there is in it, so that's why the species change. To test some of the abiotic and biotic factors, well, abiotic you test usually with devices, the biotic factors, so look at the organisms within a region. You can use two techniques. One's quadrats, and quadrats are those grid things you should have used because this is required practical to sample an area. I'm not going to take you through the whole process. If you look in your practical book, the black practical book, it's got how you do quadrat sampling. It is a random method of sampling that is used to sample the organisms living in a different area. Whereas transects are slightly different. They involve you making a line down an ecosystem or a habitat and actually sampling the organisms as you go up away from a certain area. So in this example here, you've got the sea, then the beach, then heading up to the sand dunes, then behind the sand dunes. We can see if the organisms in those different environments change as the distance from the beach increased. And we would see a pattern in that, and we can make some conclusions about that. This is a more systematic method of sampling because you're actually looking for a pattern. This you're just generally looking at numbers. You can get a math style question with this where you actually have the area of the field. It tells you how many organisms there are in a quadrat, and you have to work out how many quadrats you can fit into that area and multiply the numbers up to find the total number. So I'm going to cover sort of one last thing, and there are some more slides I didn't have time to get onto, but I'll cover competition and adaptation just ever so quickly. So competition between organisms, they can be between the same organism or different organisms. Most organisms compete for things. Plants compete for light, water, minerals which are in the ground, and space to actually grow, and they have certain adaptations which help them with that, doing it better. And animals will compete for food, territory, mates. They will need water as well to survive. They could technically compete for that, but quite often they can get a supply of that if they live in a certain environment. Similarly for plants, they do need CO2, but CO2, since it's in the air, they don't have to compete for it, they can all get access to it, and they can't get better at taking it in really. But they can be adapted to do the other things better, or to actually be a better competitor. That's what adaptations are all about. You make a plant or animal better at competing. You can get three types: structural, which are actual different structures within the organism, so different shapes. So for example, the polar bear's got small ears to prevent heat loss. That's a structural adaptation. Behavioural means how the plant or animal actually acts. The bear itself sleeps or hibernates through the winter because there's very little food about, and that's a behavioural adaptation to prevent it dying in the cold winter months. Physiological is things that the body actually does in response to a change. So that would be a spider or a snake producing venom to paralyse its prey so that it can eat it better. Physiological for a plant might be producing a toxin, or a stinging nettle producing a toxin that hurts you if you go near it, to put off animals that may want to feed on it. Good examples for that would be your polar bear, your cactus, hot environments, cold environments. Those are the sort of things you need to think about adaptations for. Okay, I'm not going to go through them all, but think about hot environments, cactus and camels, things like that. Cold environments, generally you're looking at animals such as polar bear, Arctic, like fox or hare, things like that, are the things you're talking about. We didn't get on to decay and sort of cycling of materials. There are some cycles you need to be aware of. The water cycle, if you download the slides you can see that. You need to know the main stores of it and how water is moved between them. And the carbon cycle, if they ask you questions about it, it'll be about humans who are affecting this. They're burning fossil fuels which release CO2 into the air, causing global warming. They're cutting down forests, which usually takes CO2 back in. And also when they burn forests, it releases CO2. And is a big problem. And then humans using land. As population rises, we need more land for farming, building, quarrying, dumping our waste. Often we'll cut down trees, and again that ties into the carbon cycle, because trees take in carbon dioxide through photosynthesis, causes a problem there, and can lead to global warming if there's too much CO2 in the atmosphere. And if we burn down a forest, such as what somebody's doing here, because we want this land for use, usually for farming, it actually releases even more CO2 into the air. So that's quite a quick run through towards the end. We did really good detail on homeostasis, nervous response, hormone control, and genetics. We're a bit fast through variation and evolution. Hopefully you got the main points. As I say, download the slides. I haven't covered literally everything in the course because I would need like sort of a whole night to do that in great detail. So do use other revision sources such as our website, revision guides and other things to do it. Watch this though. Hopefully you've watched it live, but if not, watch it again like when you can over the weekend. And your exam will be Monday morning, so we will be in Monday morning just to go through any questions you may have, last of all that came up in your revision. Otherwise, good night, and it's Friday, so I'm very tired, but hopefully this has been useful.